is Michael Levitt. I've been involved with Insilico medicine since 2014, shortly after I won a Nobel Prize in chemistry. It's been a great pleasure to be involved with the company because it's been moving ahead so quickly. And today I want to discuss and announce a new lead drug, a preclinical candidate for an inhibitor of coronavirus. I should stress that actually for much of the last 800 days or 820 days since January 2020, I've actually been working nonstop on corona. I even have a placemat shown here showing the pandemic as it was worldwide in September 2020. Since then, it's become a lot more complicated. But for me to be able to describe a drug that would help alleviate the effects of corona and COVID is actually a real pleasure and treat. So basically, it's remarkable a company, say without the resources of a major big pharma company, has able to actually get a drug for COVID in a very, very short time. But when you actually see what Insilico medicine has done, they really seem to have a scheme. They really seem to have a method which is working not just once, but many times to basically speed up the development of drugs. So typically, drug development is a very complicated and long process. It looks like a pipeline starting at one end with the idea of a drug and ending out with sales and marketing and real world evidence. And this typically costs a few $1 billion of development costs once you actually have the target. Insilico medicine has really devoted itself to speeding up the whole process. Now, as you can imagine, a process that involves a pipeline, in fact, several pipelines, really is no faster than its slowest step. So it's really important to make every step efficient. And this is something which has been done by Insilico medicine. They've managed to greatly speed up the times of each step, reduce the costs in millions of dollars to launch each step, and also enhance its probability of success. So this is actually something about the company. It's not something about the specific COVID drug. So if you look at traditional research and discovery for drugs in a detailed view, you realize, as I said, it's not a single pipeline. It is just a large number of different steps. Perhaps the hardest step, at least nowadays when so many drugs have been developed, is to actually have a hypothesis about a disease. Now, this was relatively straightforward for COVID-19, for coronavirus. It's a virus, try to stop the virus growing. But for many diseases, we don't actually know what is the cause of the disease. And as you'll see, Insilico medicine not only can target situations where we know what the disease is, but also target situations where we don't. And basically, just to read off some of these titles for a disease, we need to understand what's the origin of the disease, what causes it, some clinical work, trial and error, find a target. Once we think we know what the protein target is, we have to validate it. We have to see what pathways it's in. We have to understand its physiology. We have to understand how it connects to the disease. We have to do preclinical assays. Then we come to try to find a small molecule, a drug, that will interact with the protein and essentially modify its action. This is how almost all drugs work. So this requires a crystal structure of the protein, synthesizing compounds, looking at the compounds functionally, looking at absorption, distribution, and metabolism, ADME, checking toxicity. Then we have a lead. We now need to optimize the lead. The lead should be pharmaceutically available. It should be efficient. It should be metabolized okay. It should have the right doses. And after all these steps, we can finally think about filing an IND, an initial drug discovery document, which is an important step, meaning we have gone through 30 or 40 steps already, and then things start again. So essentially, you can see that it's not surprising that new drugs are taken to markets by huge enterprises that are essentially a conglomerate of many, many different independent research institutes. And Insilico Medicine. Silicon Graphics was a company I used to use many, many years ago, forgive me. Insilico Medicine's basic modus vivendi, mode of operation, is to find a way to make all of these steps more likely to succeed, less costly, and quicker. So it's by optimizing the entire network of steps that this is achieved. And this is done by a combination of intelligence, human intelligence, having the right people, being in the right places, and also using AI, artificial intelligence, to use as much data as possible. And typically, as I said, it involves not just a huge company, it involves different departments. It involves academia, often involved in validating targets, finding diseases, academic medicine. Then it involves a chemistry department who can do all the optimization and the IND enabling studies. And then finally, a clinical department that needs to test safety, formulation, manufacturing, and so on. So you can actually see that what ends up being a little pill that makes you better is a very, very difficult thing to have. Okay, you can also see this as a block diagram, not just a flow chart, where different areas are important. And again, we have in Insilico, special AI and very smart operations devoted to identifying targets. As I said, this is not a problem for COVID-19, but is a problem for novel drugs, and novel drugs are the important ones. Then finding small molecules that can inhibit the proteins identified. And finally, doing the clinical trials. And each of these steps themselves involve a whole set of optimizations. Now, another thing about Insilico Medicine is that they have two very important scientists, Alex Zaboronkov, the founder and CEO, 20 years of experience in bioinformatics, listed as one of the top 100 AI leaders, an adjunct professor at Buck Institute, publications, books, and a BSc from Queen's University and a PhD from Johns Hopkins. And he is balanced in a sort of yin-yang fashion by Feng Reng, the Chief Scientific Officer, 14 plus years of experience across pharma and CROs in both the USA and China, head of drug development, discovery and service in Medicylon, former director of GSK, PhD from Harvard University, and peer-reviewed publications. So we have somebody, Dr. Feng Reng, who is sort of an expert in the conventional pharmaceutical process. And then we have Alex Zaboronkov, who is coming in trying to break the mold from the AI point of view. And these things together, I think, are what gives Insilico Medicine its unique ability to manage all these steps as it does. So again, we have another set of blocks. And here we see, focusing on the middle area of the slide, that there are these two areas, AI and clinical research organization management, trying to understand the information, trying to get the information to give you as much as it can through AI, but also managing the efficiency of all the steps that have to be done, managing a biology team, a medical chemistry review, clinical research organization prioritization, who should do what, the preclinical team, chemical manufacturing and control team, and a formulation team. So we have these two different areas interacting, and this is what gives us the strength. But it's important to realize that they have to be very tightly coordinated, because as I said, any step that isn't happening at the right time slows everything down. This also involves working in different places. And China is certainly part of where operations happen. But Insilico medicine traditionally has worked in other places. So essentially, the idea is to work in the location that most effectively makes the process efficient. So for the COVID-19 program, a program which is recent because no one knew about COVID-19 2 years ago, 3 years ago, well, certainly 2.5 years ago, the COVID-19 program really was looking for an orally active small molecule of the 3CL like the 3C like protease for the treatment of COVID. And in a very, very short time, certainly less than 2 years, Insilico medicine has been able to find a preclinical candidate. And this is really important because when it comes to a pandemic, time is of the essence. We were lucky that some of the big pharma companies were able to devote resources and also repurpose drugs that they already had. And besides vaccines against the virus, drugs that make it less serious are incredibly important. One of the great problems with COVID that I found from my studies is that people didn't know how dangerous it was. When there was a lot of pressure, there was a worry that we were facing a global disaster at the same level as the 1918 influenza. This turned out not to be true. But had there been medications that doctors knew could be given to the most serious patients, panic would have been much, much less. Some of the early treatments were actually not really well thought out. Those of you who remember what was happening in Italy, New York, in the spring of 2020 will remember the extensive use of ventilators to help you breathe. And ventilators were made in very large numbers, but in fact, they weren't really that important. So it's very important to have a repertoire of drugs that can help deal with respiratory viruses. And this is what Insilico medicine has devoted its energy towards. So basically, this is an overview of COVID cases and deaths. One thing you immediately see is that there are multiple waves. And in my own research on this, which started in December, in January 2020, very shortly after the first announcement, I was concerned about the dynamics of a single wave. And I must say, I did not expect there to be this number of waves. But when you think about it, we needed to get a lot of people sick and actually have a large number of deaths to actually get everyone in the end immune to some extent to the disease. And this is why most recently, the Omicron variant, which is spreading very high, is not causing as much death as the earlier variants. But this is a combination of vaccination, of better clinical treatment, of less panic. People realize that the Omicron is a less serious variant. And also because Pfizer has a drug called Paxlovid, which gained emergency use authorization and very quickly reached worldwide sales that are projected to be something like $20 billion in 2020. So this is something which has been very important in dealing with the disease, but it's also been very important as an opportunity of having a drug that could really, really, really provide real value. And the Paxlovid drug is a combination of two different drugs, nirmatrelvir and ritonavir. One is a specific inhibitor of the 3CL protease. The other is a general antiviral drug. And it was pushed forward very quickly through emergency use authorization but it's a drug that actually was around. So in finding a new drug against COVID, as you will see Insilico medicine has done, is a real breakthrough. So why is a protease? A protease is a protein that cuts proteins up. So important in targeting COVID-19. Many viruses actually make their proteins in a long single piece, and then a protease, and like a pair of scissors, comes along and cuts the protein, the long polyprotein, up into individual functioning proteins. So the virus can make the protein, but if the viral protease is lacking, then these proteins will not be in any way dangerous. And the protease is special. It needs to basically know how to cut up the long string, the long viral protein in the right places. And this is a protease called MPRO, 3C-like main protease. And it is actually the target both of the Pfizer drug and also of the drug from Insilico medicine. It's difficult, it's relatively easy for a virus to make its spike protein look different, because the spike protein is just the face of the virus seen externally. It needs to escape antibodies, and all it really needs to do is to decide which cells the virus can target. And it's possible to target a cell by having slightly different faces. Changing an internal protein, like the main protease, 3CL main protease, is much more difficult, because this protease needs to be able to cut up the long chain always in the same way. And in fact, the proteases for the virus are special. They also are not like proteases in the cell. If cells had proteases that could do this, the virus wouldn't bother making one. It makes one specially because such a virus doesn't exist in normal human cells. And this means that there's a little cross-reactivity. If you can block the viral protease, you will probably not affect the normal proteases needed for good functioning of a cell. Now, in order to get drug, which is called ISM, Insilico medicine, to become a preclinical candidate, and this is something that was all done in a short time, basically involved, on this panel I count, 15 different steps. And these steps are things like novel covalent structure generated by chemistry 42. That's important because you want a drug that isn't a copy of the Pfizer Paxlovid. It's a drug that looks different. And because of the ability to generate drugs with very different scaffolding, Insilico medicine was able to start out with about 50 different scaffolds and test each one, initially in silico and then in the lab. This drug has quite acceptable solubility, it has good permeability, and really importantly, it is synthesized very easily. Again, if you start out from scratch, you can choose drugs that maybe look the same in the end, but are much easier to make. And the synthesis is really important because the Merck drug, Paxlovid, is a difficult synthesis. And this means that more chemistry needs to be done in the manufacturing. It needs to be done not just initially, but every single time. And therefore, the drug will be more expensive to manufacture. It was very important to do in vivo and in vitro pharmacology to make sure that it had activity against SARS-CoV-2. And here the IC50, which is a measure of how active it is 19 nanomolar. Getting into the nanomolar range is where everyone wants their drugs to be. It has good cellular activity against different strains, Omicron, Delta, MERS, and SARS. Again, this protease is shared by different viruses. In fact, COVID, which was seen to be a new virus, actually isn't such a new virus. Many common colds are also very weak versions of coronavirus. Coronavirus are animal viruses which seem to jump to humans every so often. And therefore, having a general set of drugs that will basically inhibit viruses is going to be really, really important. It was very important to look at in vitro and in vivo pharmacokinetics. How does that drug change when it's given? Is it soluble? Does it become a dangerous drug? Sometimes a drug molecule gets cut up by the body as a way of disposing it, and some of the cut up products, some of the side effects are actually dangerous. So we now know that for ISM 036, 076, there are no specific metabolites for humans in vitro. So none of the things that are made are metabolized by humans. It is a much weaker P-glycoprotein ligand. So there's no drug-drug concerns. It has moderate oral bioavailability across species. Again, important. Remember, it hasn't been tested in humans yet. So in any case, all these different steps have been checked off. In order to start to think about nominating a drug as a preclinical candidate involves checking off all of these steps. And this ISM, Insilico medicine, has done IMS in its way, in an efficient way. It wasn't like any step suddenly blocked everything. So again, the key thing here is to have a pathway of many steps and to know how to do each step in the most efficient way, not something that's easy. So how is different from its competitors? So basically, it's got a very favorable absorption profile. Because the Pfizer Paxlovid is a combination of two drugs, we have to worry about that. The drug has good permeability and better human bioavailability. So therefore, it's not necessary to combine the drug with ritonavir. One reason why Pfizer had to combine Paxlovid as two drugs, one was the nirmatrelvir drug, which is the real active drug, but it wasn't very bioavailable. By combining the two, the body absorbed the others. The drug from Insilico medicine doesn't affect cytochrome P45 inhibition, which could cause dangerous side effects. It has a broader spectrum of antivirus activities. In other words, it's a more specific inhibitor of the protein, probably, and therefore is able to inhibit other proteins. Remember, every drug binds to the M protease in a different way. And some drugs can be more specific for the family, but maybe not just hitting one particular variant. It has a novel scaffold with covalently irreversible pharmacore and distinct kinetics that are different in this mode. So basically, it's a molecule that doesn't change in a difficult way inside the body. All of these things bode very well for progress with the clinical trials. So basically, if we go back and look at what has been done so far using the same pathway that I think I showed on my second slide, which goes from target to hit all the way to real world evidence in sales and marketing, these many, many steps that typically take many, many years and cost many, many billions of dollars. Insilico medicine has, in a very, very short time, got through target to hit to lead, and lead optimization, and is now entering the preclinical trial, the IND. So now we have the molecule. We have to now start to see how it's likely to be in the phase 1, phase 2, phase 3 trials. So basically, by doing the discovery so quickly, Insilico medicine has placed itself in a very important position vis-a-vis the future development. And one more thing. Remember I said to you that what was really important about Insilico medicine is that they have a process. They have this process combining traditional drug development with AI. They're very, very tightly linked together, whether it's yin-yang or left hand holding right hand. There's this very tight coupling between Alex Zavornskov, the AI guru, and Reng Deng, the person who is really, really good, or Feng Reng, forgive me, Feng Reng, who is really, really good at conventional drug design. He knows what steps needed to be added. And this, in fact, has been very important for a drug which is not needed for COVID, but for a different purpose. And this is basically a drug where they went from start to phase 1 in 30 months. This is Insilico Medicine's AI-designed anti-fibrotic drug. Fibrosis is a serious problem. Fibrosis of the lung, fibrosis of kidneys. Basically, tissue is built up in response to damage, but this tissue is not really healing. It's almost like having scar tissue, which is no longer useful. And they basically were able to start with a novel fibrotic target that was discovered by looking at genomic data, by looking at what genes were expressed more in elderly people who were more likely to have fibrosis. They were able to find a new protein that had not before been implicated in fibrosis. They were able to find a novel molecule with the desired properties. Remember, they can generate a very broad spectrum of molecules, and therefore you go into the process not with a single lead, but a whole stable full of leads. And if any one lead doesn't look so good, you have backups, and that's really, really important. They can then, by this having these multiple leads, they can worry about synthesis, process chemistry. In other words, choose a lead as they did for the anti-COVID-19 lead that would be easy to make, something that is very important. They did the in vitro cell-based assays, see how it works on cells. They were able to do translational studies on people. In vivo studies, step number 6. IND enabling studies on mice. Phase 0, human studies deciding on how much dosage would be needed. And then finally, they're currently in phase 1. Patients are getting this treatment. So basically, you can already see that in this 30 months, there were many, many steps, because each little marker here, like synthesis, is not a single molecule, but it's a whole panel of molecules that are all treated together. So I think for me, this previous work, coupled with the work on COVID, very dear to my heart, shows that there's a scheme here. It's not a one-off lucky event. Insilico Medicine really seems to understand how to make drugs more efficiently, how to make drugs more cheaply, and how to make drugs that will get to market more quickly. And this is really important because the world cannot rely on the drugs that were made decades ago, the low-hanging fruit. Some drugs that we use all the time, like aspirin, were discovered hundreds of years ago. And for this reason, we need methods that can basically develop drugs very quickly. Now, obviously for COVID, this was essential. But, even here, Insilico Medicine stepped up to the challenge while they were working at the same time on the anti-fibrotic drug and other drugs designed against cancers. They still had the bandwidth as a small company to develop the COVID drug. So basically, the anti-fibrotic drug and the COVID drug really fit together, sort of back to back, encompassing a company that has real power and potential. Thank you.